Greetings! My group members and I will be presenting a microbe known as Acetobacter xylinum that is utilized in the fashion industry. Bhavya Sri Rajesh will be presenting a brief introduction of Acetobacter xylinum that is used in the fashion industry. Nimasha Dilkijayasura will be presenting its description of Acetobacter xylinum. Susan Christiana will be presenting its mechanism and history of Acetobacter xylinum. Nimra Anjum will be presenting how acetobacter is applied and its benefits in the fashion industry. Lavanya Harshini Ganasaraja will be presenting the future prospects and challenges. Nalinakshi Yogeshwaran will be presenting the conclusion.
Hi, I'm Bhavistri Rajesh and I'm here to give you guys an introduction on the topic fashion industry as well as the importance of microbes in fashion industry and also the rationale in choosing this topic for our presentation. So, mainly, what is fashion industry? Fashion industry refers to a large industry that involves wide variety of creativity, commercial ads, cultural activities that revolves around fashion and style. It also refers to the group of companies, associations, people engaged in designs, manufacturers, marketing and distribution of apparel and accessories. It includes a broad range of pursuits, careers such as fashion designing, production, retail marketing, and more. And as we all know, fashion industry always stands out for being dynamic and always changing due to shifting fashion trends, cultural influences, and customer demands. Fashion industry's importance lies in its ability to empower individuals, shape everybody's identity, stimulate the economic growth, as well as foster creativity. Okay, so now I'll be talking about the important role of microorganisms in the fashion industry. So as we know, microorganism plays a significant role in fashion industry, particularly in the areas of textile production, dyeing, sustainable practices and much more. Number 1, textile production. In textile production, microorganisms are employed in the manufacturing of some textiles, such as fermented materials. For instance, cellulose based compounds known as the microbial cellulose are produced by using bacteria called Acetobacter xylinum. These microorganisms generate cellulose film, which may be harvested to use and create fabrics in distinct qualities. Number 2, natural dyeing. Natural dyeing processes are used in natural dyeing, which are aided by some microbes. These pigments are some bacteria and some fungi. For example, the fungus Penicillium propagandum, which makes the purple pigment, and the bacteria Streptomyces collicula, which creates the blue pigment known as indigo dye. By providing sustainable alternatives to synthetic dyes, these natural dyes lessen the dyeing process negative impacts on the environment. Number 3, biofabrication. Microorganisms are used in biofabrication, which is a process that uses the living things to create textiles and other materials. In order to manufacture proteins that can be spun into fibers and utilized in the manufacturing of textiles, such as spider silk proteins, and this necessitates employing microbes. The creation of environment friendly and sustainable fabrics with distinctive features is also possible with biofabrication. Number 4, sustainable practices. In the fashion sector, microorganisms are involved in a number of sustainable practices. For example, certain bacteria that have the ability to degrade organic waste products such as textile and natural fiber waste through processes including composting biodegradation. This helps to promote circular economy principles and lessen the negative environmental effects of textile waste. Antibacterial finishes. Additionally, microorganisms are used to create antibacterial textile treatments. Bacteriocins, an antibacterial chemical produced by some bacteria, can be put into textiles that prevent the growth of bacteria and fungi. Sportswear, medical fabrics, and socks are just few examples of sectors where this application is important.
Acetobacter belongs to the domain Bacteria, specifically the class Alpha Proteobacteria. It's a rod-shaped bacterium with a rigid outer layer made of peptidoglycan. This bacterium also possesses a flagellum for motility. One of the notable characteristics of Acetobacter is its ability to produce bacterial cellulose. It is an aerobic bacterium that converts the ethanol into acetic acid through aerobic fermentation. Acetobacter xylinum thrives in a slightly acidic environment with a pH range of 4 to 6, and it requires a temperature between 25 degrees Celsius and 30 degrees Celsius for optimal growth. Acetobacter xylinum plays a significant role in the fashion industry. Its ability to produce bacterial cellulose opens up new possibilities for sustainable fabric production. Let's explore its roles in association with humans in more detail. Production of bacterial cellulose. Acetobacter xylinum is utilized in the fashion industry due to its unique ability to produce bacterial cellulose. This cellulose can be shaped into various forms and it's used as a sustainable alternative to synthetic materials in clothing manufacturing. Biodegradable fabrics. Bacterial cellulose produced by Acetobacter xylinum enables the creation of biodegradable fabrics. Now, these fabrics can be composted, reducing the environmental impact of the fashion industry and mitigating the accumulation of non biodegradable waste. Improved sustainability. By harnessing the potential of Acetobacter xylinum and bacterial cellulose, the fashion industry can enhance its sustainability practices. It reduces the dependence on resource intensive materials like cotton and synthetic fibers, contributing to a lower carbon footprint associated with textile production.
Hello, I'm Susan Christiana, and I'll be talking about the discovery, how it is applied, and the mechanism of action exerted by acetobacteria. So first, before we jump in, acetobacteria produces bacterial cellulose, which can be used in the fashion industry. Dating back, the ancient Chinese were actually the first ones to find this, where they made and called it kombucha tea mushrooms. However, the first scientific recordings of this bacteria were done by Ferdinand Kohn in 1875, where he described a filamentous organism in a concretion from a human lacrimal duct. He named it as Streptothrix foyestri and later named it as CO. Later on in 1878, Haas also found the same bacteria from bovine lumpy jaw, and he named it as Actinomyces bovis. Later on, Brown in 1886 was the first one to write a scientific paper on bacterial cellulose, describing a fermentation substance referred to as vinegar plant. He also noticed that the entire surface of the liquid was covered with a gelatinous pellicle. We'll come to what a pellicle is. Later on in 1954, Hestrin and Shukram further found that the resting and lyophilized cells in Acetobacter, another name for Acetobacteria, synthesize cellulose in the presence of glucose and oxygen. Okay, Acetobacter xylinum, another name for Acetobacteria, is found to synthesize massive amounts of extracellular cellulose in the form of a pellicle. This pellicle is then dried to create bacterial cellulose sheet. This can be used as an alternative textile. Okay, let's see how Acetobacter xylinum works. The synthesis of cellulose by Acetobacter xylinum is a 4-step process. The first step is the phosphorylation of glucose to glucose 6-phosphate using glucokinase. The second step is the conversion of glucose 6-phosphate to glucose 1-phosphate using phosphoglucomutase. The third step is the conversion of glucose 1-phosphate to uridine diphosphate glucose using UDP glucose pyrophosphorylase. And finally, the fourth step is the conversion of uridine diphosphate glucose to bacterial nanocellulose, which is the bacterial cellulose, using cellulose synthase. The bacterial cellulose is the pellicle, which is then dried to make the sheet. Okay, let's take a closer look at the process. Acetobacter xylinum secretes cellulose. We just saw how cellulose is made. It secretes it as microfibrils. These microfibrils form a row of pores along the longitudinal axis of the cell, as you can see in the diagram. They then intertwine to form ribbons of cellulose, making a cellulosic mat on the surface of the medium. This is the pellicle, which is then dried to make the sheet. The microfibrils have a tensile strength equivalent to steel. This makes it quite a good naturally made material which can withstand a good amount of tension and pressure.
Hi everyone, I'm Nimra and I will be talking about how Acetobacter xylium is used in the fashion industry. Acetobacter xylium is a non-pathogenic, non-hazardous bacterium that is utilized to produce cellulosic nanofibers. These nanofiber is similar to cotton. Then it is processed to form a dense structure called MNW, microbial non-woven, that has high density, high purity, and shape retention. After many days of process, it forms a microbial cloth on the surface of wine. Surprisingly, from where the wine enter, that can be a question of you all. To understand the manufacturing process where we use a wine, let's move on to the next slide. Let's understand the manufacturing process in detail. Actobacter xylem culture and a sterilized medium which contains glucose, peptone, yeast extract, Na2HPH4, citric acid, and distilled water is added into a wine. But before that, a wine go through the process of filtration where a vacuum pump is used to remove any biological contamination. After that, a mixture containing Actobacter xylem culture, sterilized medium, and a wine is poured into a container. Container depends on the size of garment we want. It is being placed in the container for about 20 to 30 days at about 30 till 40 degrees Celsius. After that, a fiber production will happen. Ectobacter black rod synthesizes large quantity of microfibrils, lean lines of pure cellulose. That means a cellulose nanofiber is being formed. That means a cloth is being formed. The microbial cellulose is then shaped, curved to produce a three-dimensional garment. This is a practical example of how a garment was created by wine fermentation. In the first diagram, they're harvesting microbial cellulosic nanofiber from the cultured medium, as talked about in the previous slide. Then they are drying it and shaping it. For giving it a variety of color, a controlled nutrition can be used. And tantana, original wine dress is ready. This dress was first made in 2007. You all might be wondering that if we are using a bacteria to manufacture a garment, wouldn't it be harmful for human? Well, let me tell you, Actobacter xylem is beneficial for human. It eliminates germ transfer from the wearer to the sleeve by cuffing. It has a virus fighting capability. New research shows that denim jeans disables over 99% of viral activity within 2 hours of contact. It has an odor control capability where it reduces development of unpleasant smells and prevent the growth of odor causing bacteria. Dress fabric inhibits hosting bacteria and viruses including COVID-19. It lowers the toxicity risk and beneficial in high contact environments such as hospital where exposure of getting a bacteria and virus is very high.
Hello, I'm Lavanya. So, everyone else has already explained how Acetobacter xylinum works and explored its uses. So, I'm going to continue on by elaborating what's in store for the future of this bacterium in the next 10 to 15 years. I'm also going to be talking about the challenges that will be faced in implementing this bacterium at a full scale in the fashion industry. So, the main key prospect is that it is a keystone in sustainable fashion. So, the advantages of using fabric derived from Acetobacter xylinum is number one, it's biodegradable, number two, it uses sustainable farming methods, and number three, it's renewable. So, from now on, I'm going to be referring to the fabric as bacterial cellulose or BC, just for the sake of it not being so wordy. With regards to biodegradability, cellulose materials, BC included, are made of carbon, oxygen, and hydrogen. It can be decomposed by other bacteria and fungi in the soil using cellulases. So, why this is very important is because it can be decomposed when buried in landfills, hence, significantly reducing landfill and soil pollution. It's definitely not something new that both of these forms of pollutions are big problems in the textile industry since majority of fabrics are made of synthetic fibers like polyester and it is not biodegradable. Secondly, with sustainable farming methods, it can be compared to and even considered an alternative from cotton, which is the main material used in making garment fabric. However, cultivating cotton is resource extensive in three ways. Number one, it requires large amounts of land to be cleared for farming. Number two, cotton is a water intensive plant, which means it needs huge amounts of water to grow optimally. And it's around an estimated of 20 inches of rainfall. In areas where there is a lack of rainwater, water must be farmed from underground or from external bodies like the ocean. Thirdly, pesticides are used, and chemical pesticides are preferred over organic ones. This can seriously pollute waters and the soil. Lastly, in terms of renewability, the mechanism of producing cellulose does not require primarily non-renewable energy, which is so much better than synthetic polymers that needs petroleum, which is we know for now, which we definitely know is a non-renewable energy to create. So, as mentioned in the beginning, I'd also talk about the challenges in making BC as a widespread use in the fashion industry. So, there are three notable challenges. First being the price point, second being the durability, and the third is the need for pre-treatments. So, I'll start off with the price point now. The low productivity of Acetobacter xylinum means that not much BC can be produced from a strain. This is important because in BC production itself, cultivating the medium would take up an estimated of 30% of production costs, which means there's only 70% left for things like refining the medium, shaping, stitching, dyeing, etc. And this would end up making BC much more cost heavy to produce. And let's be real here, a lot of companies would rather cut corners than to spend that little extra in making something that is a little bit more sustainable. The challenge here is to identify a new low cost media that would not only produce a high yield in a short amount of time, but it would not sacrifice sustainability and quality. The next is the topic about durability. Now, in durability tests, it is shown that even though BC is more or less like animal leather, it does require a little less force to rupture. So, this means that at the current stage of research, BC is better suited for loose fitting garments rather than tight fit ones. And then in moisture transfer tests, which shows how fast moisture can transfer from a surface out of the fabric and to the surroundings, it shows that BC is the least breathable. That means transfer of moisture from the source, like say the skin, to the surroundings is the least efficient. This makes BC not very suitable to be worn against the skin or in wet climates. Now, I think for us here in Malaysia, this is definitely something that's very important because of our hot and humid climate, and we end up sweating a lot. If we wear something that's not very breathable, it's just not going to be comfortable. I mean, if you wear leather out in public and like you're just not going to feel very comfortable because your body just can't breathe very well through it. Last is the use of pre-treatments, and this is a standard procedure that is done to enhance the quality of the pulp before it can actually be used. Now, this is to increase the yield of the cellulose from hydrolysis of the natural biomass. And the challenge here is to demonstrate which pre-treatment methods can not only effectively change the structure, chemical composition, and hydrolysis rate of the biomass, but to also increase the yield, which can then solve the price point problems. It's also very important to mention that some of the pre-treatment chemicals may not be sustainable enough. It may not be good for the environment, which then dampens any claim that BC is a sustainable approach. However, It is still very important to mention that this research is still very new, which means there's much more for science to bring in the prospects of making this research much more environmentally friendly and safe for the workers and the consumers. So, with that, I've talked about the future prospects in using Acetobacter xylinum for making clothing, as well as certain challenges that would have to be encountered so that it could be a widespread use for a better industry. Thank you.
I am Nalina and I'll be concluding the presentation. In conclusion, a humble bacterium like Acetobacter xylinum should not be disregarded easily. Over the years, many species of Acetobacteria have been discovered. These species have been useful in various fields for a long time. But what sets this specific species of Acetobacteria apart is its ability to synthesize bacterial cellulose in the unique form of a pellicle. As mentioned earlier, the cellulose sheet produced from Acetobacter xylinum is a sustainable alternative to common textile materials. Not only is the textile material produced biodegradable, it is also possible to make it colored by manipulating the nutrition. The synthesis of the bacterial cellulose itself may seem complicated, but really it is just a process of converting glucose to bacterial nanocellulose. The materials required to manufacture textile from microbial cellulose is somewhat similar to what is used in the wine industry. This makes it easily adaptable to be synthesized in most countries out there. Besides, its flexible growth conditions makes it easier to be cultivated, where additional environmental changes may not be required. Properties like its resistance to viruses and low toxicity also suggests that it could be integrated into the industry to cater for various socio-economic fields. All things considered, the process of incorporating Acetobacter xylinum into the fashion industry must be researched further to ensure promising results in all aspects.